Good morning, everyone, and thank you for being here today. I'm pleased to welcome you to this presentation. Today, we will be discussing CAPA for significant changes in test results during stability studies. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this session, we will explore how to identify, investigate, and address significant test result changes that can occur during stability testing. I look forward to sharing insights and best practices with you. Let's get started. Let's start by clarifying the main objective of this slide. We aim to define what CAPA is, specifically in relation to significant deviations we might observe in stability data. It's important that we understand how CAPA fits into managing these deviations effectively. Next, we want to ensure our approach is aligned with the relevant regulatory frameworks, such as ICH Q1A, Q9, and Q10. These guidelines provide the foundation for how we handle deviations, implement CAPA, and maintain compliance across our processes. We will also look at practical examples and actions. Real-world illustrations help us grasp how to identify deviations, investigate root causes, and implement effective corrective and preventive measures. Finally, all of this ties back to our overarching goal, supporting robust product quality decisions. By establishing a clear, compliant, and practical CAPA process, we ensure the quality and safety of our products are maintained at the highest standards. Let's take a closer look at the guiding standards that underpin our approach to quality. First, we have ICH Q1A, R2, which outlines the fundamental principles for stability testing, ensuring that our products remain safe and effective throughout their shelf life. Then there's ICH Q9, focusing on quality risk management. This helps us identify, evaluate, and control potential risks in our processes. ICH Q10 emphasizes the pharmaceutical quality system, with a particular focus on corrective and preventive actions, or CAPA, to continually improve our systems. Additionally, regulatory bodies like the FDA and EMA expect us to conduct timely investigations and implement preventive measures promptly. The key regulatory intent here is clear. CAPA must be systematic, well-documented, effective, and based on a thorough risk assessment. This ensures that we're not just reacting to issues, but proactively managing quality. Now, when do we trigger a CAPA? It's initiated when we observe test results showing significant changes beyond predefined specifications. For example, if we see out-of-trend data or unexpected degradation patterns, that's a red flag. Recurrent failures across batches signal a recurring issue that needs addressing. And of course, once we identify the root cause, we must implement corrective and preventive measures to prevent future occurrences. Overall, these standards and triggers are all about maintaining a robust quality system that's proactive, risk-based, and well-documented to ensure the highest product quality and patient safety. Now, let's take a closer look at the CAPA lifecycle, which is a simplified but essential process in managing quality issues. It all starts with detection. That's when we observe a significant change or problem that needs our attention. Once we detect an issue, the next step is evaluation. Here, we assess the severity and impact of the problem to understand how critical it is and what areas are affected. Following evaluation, we move into investigation, where we perform a root cause analysis. This step is crucial because identifying the underlying cause helps us prevent similar problems in the future. After pinpointing the root cause, we implement corrective actions to fix the existing issue, ensuring that the immediate problem is resolved. But we don't stop there. Preventive actions are then taken to avoid recurrence, aiming to strengthen our processes and reduce future risks. To ensure our efforts are effective, we conduct verification and effectiveness checks, confirming that the corrective and preventive measures are working as intended. Finally, we document everything thoroughly and close out the CAPA. Proper documentation ensures accountability and provides a record for future reference, completing the cycle. By following this life cycle, we create a proactive approach to quality management, continuously improving and maintaining high standards. Let's look at this example attribute related to assay failure. So here, the key observation is that the assay dropped below the specified limits at the 6-month mark. For instance, it showed a result around 93.8%, whereas the acceptable range or specification is between 95 and 105%. This kind of deviation is significant because it indicates that the assay isn't meeting its required performance criteria. When we see something like this, it triggers a CAPA, or corrective and preventive action, process because the result is out of specification. In response, the initial actions taken are quite straightforward but essential. First, retesting is performed to verify whether the initial result was due to an analytical error. Sometimes, the issue might be with the testing process itself rather than the actual sample. Second, affected stability samples are quarantined. This step ensures that any potentially compromised samples are segregated and do not influence further testing or decision-making. Overall, this example underscores the importance of closely monitoring assay performance, promptly addressing out-of-specification results, and taking immediate corrective actions to maintain product quality and compliance. Now, let's delve into the assay failure CAPA investigation process. When we encounter an assay failure, the first step is to thoroughly investigate the potential causes. It's important to evaluate all possible factors that could have contributed to the failure. For example, we look into whether there was any drift in the analytical method or if there was a calibration lapse that might have affected the results. Storage conditions are also critical. Any excursions or deviations in storage temperature or humidity can impact assay performance. Additionally, we consider the possibility of interactions between the formulation and packaging materials, which could alter the assay outcomes. To support this investigation, we rely on specific tools. Trend analysis helps us identify patterns or shifts over time that might indicate underlying issues. We also review stability chamber logs to check for any anomalies during storage periods. And of course, we follow established protocols for out-of-specification or out-of-trend investigations, which guide us through a systematic review of the data. Connecting this to our overall quality assurance efforts, these investigations are vital for maintaining product integrity and ensuring compliance. By thoroughly evaluating these potential causes with the right tools, we can identify root causes and implement effective CAPA measures to prevent future failures. Now, moving on to the corrective actions we plan to implement. First, we will retrain our analysts on the method steps to ensure everyone is aligned and following the correct procedures. Additionally, we will recalibrate our instruments, such as the HPLC, to maintain accuracy and consistency. Updating our system suitability criteria is also essential, so we can better detect deviations early. All these actions will be documented thoroughly in the analytical investigation report and training records for traceability. On the preventive side, we aim to strengthen our method performance checks to catch issues before they impact results. We're also adding extra control samples during stability runs, which will help us monitor the process more effectively. Moreover, we will review and update our stability protocol acceptance criteria to ensure they remain robust and relevant. The ultimate goal here is to prevent any future drift below our specified limits, maintaining the integrity and reliability of our assay results. These combined corrective and preventive actions are part of our continuous effort to improve quality and compliance across our processes. Now, let's look at an example of a degradation product rise. What we observe here is that the impurity actually crosses the acceptance limit earlier than what we initially predicted. This is a key indicator that something might be affecting the stability of the product sooner than expected. Our CAPA focus in this situation is to assess the underlying cause of this early rise and to implement measures to mitigate the formation of this impurity. Essentially, we need to understand what's causing the degradation and find ways to prevent or slow it down. When we investigate these kinds of issues, there are several considerations to keep in mind. For instance, interactions with excipients can sometimes promote degradation. Also, failures in packaging barriers, like oxygen or moisture barriers, can allow environmental factors to accelerate impurity formation. Temperature and humidity also play a significant role. Fluctuations in these conditions can impact the stability of the product over time. Connecting this back to our overall theme, the key takeaway is that early detection and thorough investigation of these degradation patterns are crucial. They help us improve product stability, ensure quality, and ultimately safeguard patient safety. Let's take a closer look at the degradation CAPA, or action plan. First, for corrective actions, we plan to adjust the formulation by adding a stabilizer, which will help prevent degradation issues. Additionally, we aim to improve the packaging barrier to better protect the product from environmental factors that could cause deterioration. On the preventive side, we're increasing the sampling frequency at identified risk points. This means we'll be checking the product more often in areas where degradation is more likely to occur, allowing us to catch issues early. We're also updating our risk assessment in line with ICH Q9 guidelines to ensure our approach is current and comprehensive. Finally, to measure the success of these actions, we'll monitor the next 3 stability cycles. This ongoing review will help us determine if our corrective and preventive measures are effective in maintaining product quality over time. Let's take a closer look at this example of a dissolution drop. As you can see, the observation here is that the dissolution falls below the Q value when we look at long-term conditions. This indicates that our product isn't meeting the expected dissolution criteria over time, which can be a sign of stability issues or process variability. To address this, we follow CAPA steps, starting with evaluating the manufacturing process variability. It's crucial to understand if any changes or inconsistencies in the process could be causing this drop. Next, we check the dissolution apparatus and media quality, because equipment or media issues can also impact dissolution results. Additionally, assessing blend uniformity helps us ensure that the active ingredient is evenly distributed, which directly affects dissolution consistency. Once we've identified potential root causes, we implement actions. These include tightening process parameters to reduce variability and enhancing in-process controls to monitor critical factors more closely. The goal here is to stabilize the process and prevent future dissolution issues, ensuring our product maintains its quality throughout its shelf life. Overall, this example highlights the importance of thorough investigation and targeted actions in maintaining product performance, especially under long-term storage conditions. Let's take a closer look at the documentation requirements for CAPA. Each CAPA must include a clear problem statement that explains what the issue is and when it occurred. This helps us define the scope right from the start. Next, we need an investigation summary, which involves reviewing the data and analyzing it thoroughly to understand the root of the problem. Then, it's essential to draw a solid conclusion about the root cause. This is the basis for determining the appropriate corrective and preventive actions. Speaking of which, every CAPA should specify what actions are being taken to address the issue and prevent future occurrences. After implementing these actions, we conduct an effectiveness check to verify whether they have resolved the problem effectively. The results of this check should be documented clearly. Additionally, it's important to cross-reference these CAPA reports with stability reports to maintain consistency and traceability. All of these elements are aligned with regulatory audit expectations, ensuring our CAPA process is comprehensive and compliant. By covering these points thoroughly, we reinforce our commitment to quality and continuous improvement across the organization. Let's talk about CAPA effectiveness monitoring. To verify that our corrective and preventive actions are truly effective, we look at several key approaches. First, we review subsequent stability data points to see if the issue has stabilized over time. This helps us confirm that the corrective actions are working as intended. Next, we check related batches across the entire product lifecycle to ensure that the problem doesn't recur in different batches or at different stages. We also use statistical trending, like control charts, to monitor process stability and identify any patterns or deviations that might indicate ongoing issues. These tools provide a more objective view of whether our actions are truly effective. Now, moving to key metrics. Two really important ones are the recurrence rate of similar deviations, which tells us how often similar issues happen again, and the timeliness of CAPA completion, because the sooner we close these actions, the less risk there is of further impact. Overall, these methods and metrics help us ensure continuous improvement and maintain high-quality standards across our processes. So let's take a moment to review the key takeaways from today's presentation. First, corrective and preventive actions, or CAPA, are absolutely essential when it comes to managing significant changes in our processes or systems. They help us address issues effectively and prevent recurrence. Next, it's crucial that our CAPA efforts are driven by root causes. We need to dig deep to understand the underlying issues, rather than just treating surface symptoms. This approach makes our actions more preventive and effective in the long run. Third, robust documentation and trending are vital components. Keeping thorough records and analyzing trends over time enable us to identify patterns and potential risks before they escalate. This ties directly into maintaining a strong quality system. Finally, regulatory authorities expect us to be proactive in our approach to quality management. They want to see that we're not just reacting to problems, but actively working to prevent them through a well-designed, proactive quality system. All these points really connect to our overall goal of ensuring product quality and compliance. By focusing on these areas, we strengthen our processes and build trust with regulators and customers alike. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Feel free to contact us through the provided channels. And finally, I want to sincerely thank you for your time and attention today. It's been a pleasure sharing this information with you, and I hope you found it valuable. Remember, we're here to support you beyond this presentation, so don't hesitate to get in touch. Thank you again.